Good morning, good afternoon, good evening. Welcome to Global IMMUNOTALKS. It is my great pleasure to introduce today's global immuno speaker, Professor Adrian Hayday. Adrian is the key Glendinning Professor of Immunobiology at King's College London and a principal group leader at the Francis Crick Institute. He studied biochemistry at Cambridge and earned his PhD in tumor virus genetics at Imperial College London before moving to MIT, where he co-discovers genes defining the gamma delta T cell commitment. Together with Susumu Tonegawa, he opened an entirely new area in immunology. He then started his independent career at Yale, where he rose through the ranks to professor, and in 1998 he moved to King's College London as the founding chair of the Department of Immunobiology. During his time at Yale and later in London, Adrian discovered that gamma delta T cells are uniquely specialized for tissue surveillance, demonstrating that mice lacking these cells are highly susceptible to skin cancer and showing how gamma delta T cells discriminate between healthy and stressed or transformed cells. His work has provided a framework for understanding how the immune system senses normality and has opened therapeutic avenues for cancer and inflammatory diseases. Adrian's many honors include the Devan Medal. This medal is given to members of Yale faculty that have distinguished themselves as teachers of undergraduates in Yale College. Adrian has also been elected as a Fellow of the Academy of Medical Science and the Royal Society. I very much look forward to his lecture today entitled Lymphoid Stress Surveillance, Normality Sensing, and Integrated Immunology. So, Adrian, as you know, one of the traditions of Global IMMUNOTALKS is to ask a special question before you start your talk to inspire the young generation of scientists. So the question for you today is, can you describe a turning point in your career that defined your current research focus?
Yeah, well, first of all, Gabby, thank you very much for the kind introduction, and thanks to the team at Global IMMUNOTALKS for organizing this with their customary efficiency. It's a real pleasure and honor to be with you. In answer to the question, it was 1997, and I was just turned 40. And you're right, I'd been working on gamma delta T cells, but it was difficult. It was really difficult. There was no frame of reference. And we had a lot of work going on T cell development at the time, thymopoiesis, and it was going well. And so I really thought, maybe I'm going to give this gamma delta T cell stuff up. And I was at the Society of Microbiology meeting in 1997 in Miami Beach, and there were about 8,000 people there. And suddenly I saw Peter Doherty, who had just won a Nobel Prize the year before in immunology. And everybody works in silos, so no microbiologist had any idea who this guy was. But we saw each other and we yelled to each other. We went out for dinner that night in Miami Beach at a restaurant where Peter pointed out that everybody was much prettier than the two of us were. And we just chatted and chatted, and he just said to me, Adrian, you can't give this up. These are conserved cells. There's a biology here that has been conserved for hundreds of millions of years. We've got to know what it means. And from that moment on, I realized that that's what I had to do. So it's thanks to Peter Doherty for doing that.
Wow, this is wonderful. Thank you so much, Adrian, for these very inspirational words. And now I would like to invite you to share your screen and deliver your lecture.
Great, thank you. So I'm assuming that can be seen.
Yes, perfect.
Good. Let's just get the laser pointer up. Okay, so once again, let's just get this here. Yeah, that's it. Thank you for the invitation. The work I'm going to describe today has been done at these two institutes, King's College London and the Francis Crick Institute, as you correctly alluded to. I have some disclosures that are shown here, but none of these entities have influenced in any way the content of this talk or have sought to influence it. I'm going to give the acknowledgements right at the beginning. It's probably the most important thing that I can do. The work includes some considerable unpublished work that was really the brainchild of Miguel Munoz, who's now running his own lab in Madrid. But he continues to work with a postdoc from our team, Nicolas Villand. And there's also work from Cara Brown, our lab manager, assisted to a considerable degree by Duncan Berger, our bioinformatician. There'll also be some work from Dasha Fredina and Ambra Nattalini, but also thanks to sort of everyone in the lab, the support that we are provided with, and also to the science and technology programs that facilitate so much of what we do. So with those acknowledgements stated, I want to consider that, whoops, let's get this, there we go. That applied immunology predated studies of immunology. And of course, what I'm considering is that as long ago as 1796, Jenner really began the process of modern-day vaccination as we understand it, building on a centuries-old tradition of variolation, whereby inoculation with small amounts of a potentially pathogenic agent could elicit protective immunity that was long-standing. And we know that vaccination has saved the lives of untold millions of people and improved the quality of lives for at least as many. And it is an unconscionable, tragic, and utterly unjustifiable fact that it is under the kind of attack that it now is, which reminds us all that it's not just about science, that science have to be responsible as a community, as exemplified by global IMMUNOTALKs, to make sure that reason and fact and scientific endeavor are not subject to the kind of unjustified attack and criticism that they're suffering today. The success of vaccination led to the studies of immunology through individuals, of course, such as Pasteur, but later on by many others. And what emerged was a complex system that balanced the capacity to make immunogenic reactions that protect us against a balance of maintaining tolerance so that we did not suffer what Paul Ehrlich referred to as horror autotoxicus, the capacity of these immensely diverse immunoreactivities to react against self. And it was recognized by a number of individuals, but perhaps best articulated by the late Charles Janeway, a former colleague of my own, that this immunogenicity versus tolerance was determined not by the specificity of the lymphocyte, but by the integration of what we now term innate and adaptive immunity. So this will be familiar to many of you, but the notion, which has been shown through durable experimental studies, is that there are antigen-presenting cells which have innate receptors. This would be a toll-like receptor related to those first identified in Drosophila, that is capable of recognizing pathogen-associated molecular patterns and thereupon upregulating molecules of the B7 superfamily that provide contextual information that will allow a quiescent T cell to be activated. Excuse me. What you see here on the left-hand side is the skin, the epidermis of a mouse, and the cells there are the anatomical manifestation of this integration of innate and adaptive immunity that I just described. These are the Langerhans cells brought back to our attention by Rolf Steinman. And these cells are indeed capable of sitting in the tissue and becoming activated by these pathogenic signals and thereupon passing both antigen-specific and contextual information to T cells. This constitutes a rapid communication of contextual information that is essential for immunogenicity. But things are, of course, more complex. If you look at the skin, as I just showed you, of a mouse, those Langerhans cells, now shown in cyan, are seen to be not the only dendritic cells that are occupying that space. There are equally large numbers of these dendritic cells, shown in red, that are actually T cells, not surprisingly termed dendritic epidermal T cells. They have a monomorphic gamma delta T cell receptor. It happens to be encoded by a V gamma 5 chain and a V delta 1 chain. And if you look in detail, now you find that 70% of these T cells at steady state are in contact with the Langerhans cells. A mere 30% showed in yellow at any one point in time are not in contact. This intimate relationship of these gamma delta T cells with Langerhans cells has, to my knowledge, not been resolved in functional terms by anyone to this date, but it gives us a clue that sensing the status of the tissue is not simply going to be the province of these Langerhans cells, but is going to be contributed to by other players. And in that includes what we're going to term sentinel lymphocytes. So what do these gamma delta T cells do that are sitting in the epidermis of the mouse? Well, they and other tissue intrinsic gamma delta T cells have been implicated in cancer immune surveillance, initially from the work that we published about a quarter of a century ago now, but more recently through human studies showing in a number of situations, lung cancer, breast cancer, ovarian cancer, beneficial roles played by gamma delta T cells in immune surveillance, all of which was reviewed by myself and my close colleagues just about a year ago. So if they're involved in this kind of surveillance, what is the underpinning mechanism? And there's a cartoon here that was put together by Pierre Vontereux, a postdoc in our lab, and myself some years ago, that shows that stress to an epithelial surface will upregulate molecules such as this one here, Ray1. This is a sister molecule of a well-known entity called MICA. And it is recognized by an innate natural killer receptor, NKG2D, and that's capable potentially of inducing a response in these T cells. What the T cell receptor is doing during all of this is unclear, and the question was addressed by Jessica Strid, shown here, when she was a postdoc with us about 15 or so years ago. She built a bitransgenic system in which we could induce with a molecular switch the upregulation of this molecule Ray1, which is ordinarily stress-induced. And what she showed was that that was sufficient to induce the rounding of these red cells, which are the gamma delta T cells, but it also induced the rounding of the blue cells, which are the Langerhans cells, which do not have a receptor for Ray1. The T cell activation was manifest by an upregulation of CD69. The Langerhans cells upregulated CD86. So this seemed to show that these T cells sitting in this epithelial surface had simply innate responsiveness to stress within the epithelium. And of course, the time frame for this can be in hours instead of the several days that we think of T cells ordinarily responding in. So that enabled us to build this hypothesis of lymphoid stress surveillance, wherein stress is now detected on the local epithelium by these local T cells using innate receptors in a polyclonal or non-clonal fashion, transmitting information to dendritic cells that we subsequently showed will also prime antigen responses of T cells. So now again, we have a rapid communication of contextual information via the innate seemingly to the adaptive, but it's mediated by lymphocytes rather than being initiated by dendritic cells. There's another example of this. It's also relating to the skin, but it relates now to gamma delta T cells which sit in the dermis. They have a slightly different T cell receptor for those who are the cognoscenti of this, V gamma 4 or V gamma 6 instead of V gamma 5. But the point is if you simply apply imiquimod or Aldara, which contains TLR7 ligands, to the skin of mice, it will induce a rapid psoriasis-like pathology within a very small number of days, associated with local swelling of the skin. And this has been attributed to the production locally of IL-23 and IL-1 beta, which activate these dermal gamma delta T cells, which make IL-17, which induce neutrophil attraction and stimulation, leading to infiltration of the skin. Again, this is a rapid communication of contextual information by tissue intrinsic sentinel lymphocytes. But again, any role for the T cell receptor is unresolved. In fact, in this diagram that you can download from the literature, the T cell receptor isn't even shown as a molecule on the surface of these cells. So this question needs to be answered, because if you don't need a T cell receptor, why do you even need T cells to make these innate responses? There are many other cell types, innate lymphoid cells, NK cells, that would be able to do this. So we ask, what role, if any, does the T cell receptor play in this process? So what you see here is another depiction of these dendritic epidermal T cells. And what you can see here is that the T cell receptors are actually clustered in areas of high density, these projections. And when you unmask the epithelial cells in this visualization here, you realize that these projections are all aligned with the surface of the epithelial cells. And Thomas Schall several years ago showed in a beautiful paper that these T cell receptor projections are coincident with steady state signaling as demarcated by the accumulation of phosphotyrosine. So what does this signaling in the tissues from the T cell receptor reflect if the T cell receptor is really not doing anything, as these models of innate immune surveillance seem to suggest? So to address that, Miguel Munoz-Ruiz, and now with Nicolas Villand, chose to target a component of the gamma chain of the T cell receptor, the C gamma 1 gene, which is expressed by all gamma delta T cells that sit in the tissues of a mouse. So V gamma 5 in the epidermis, V gamma 6 and V gamma 4 in the dermis, V gamma 7 in the gut, etc., etc. And what they said is, let all of these repertoires develop, and then when they've developed, we'll apply tamoxifen to delete specifically this receptor component, specifically in gamma delta T cells. Excuse me. There are two types of mice that we can consider here. Both of them have this inducible Cre excision machinery active, but in one of them, the T cell receptor C gamma 1 gene is not floxed, so it remains intact. In the other one, it's floxed, so it's removed. If you look in the epidermis at what happens, CD45 demarcating these lymphoid cells in the epidermis are still there following T cell receptor deletion. But of course, the T cell receptor staining has gone because, as you'll see in a moment, this is a high specificity, high sensitivity system. You can actually find relatively low levels of CD3 staining in these cells, but this is not because we've got alpha beta T cells coming in. This is all within just a week of removing the T cell receptor. Rather, the T cell receptor proficient cells that normally have very high levels of CD3, upon loss of the T cell receptor chains, the CD3 becomes greatly reduced in expression, can be detected intracellularly, but at small levels on the surface as well. However, these cells are completely non-responsive to any anti-CD3 activation. So essentially, the functional response through the T cell receptor pathway has been abruptly ablated, prior to which everything was normal, and now it's acutely removed. By removing the T cell receptor, the problem is we've removed the signature marker for these cells. This was resolved by crossing the mice to mice known as MTMG. Many of you may have used these. And in these mice, in cells where there is Cre activity, a tomato a marker, fluorophore, is converted to GFP. This works extremely well. Before there's any Cre activity induced by tamoxifen, there are simply no GFP positive cells. When we switch on tamoxifen, was when we administer tamoxifen and switch on the Cre. Within a short period of time, GFP positive cells accumulate, over 99% of which were gamma delta T cells in the epidermis. And likewise, if you look at all of the T cells that were in the epidermis, over 90% of them have become GFP positive. Only a residual, about 10% remain GFP negative. So this is a system that's both specific and sensitive. And we can also look in the dermis because, as I mentioned, those cells too use this particular gene segment. And although the data are slightly lesser, nonetheless, it's very compelling. About 89% of the GFP positive cells were previously gamma delta cells. And those of the gamma delta cells, 80% have become converted to GFP. So now we can track these cells, and much to our surprise, over 2 months after TCR deletion, these cells are still here. There might be a trend to slightly declining numbers, but it's not significant. That's in the epidermis. And likewise, there are comparable numbers between 1 to 2 months in mice which retain the T cell receptor or in mice which have lost the T cell receptor. So these epidermal and dermal cells are still there. And now we can study them and see what happens to them in the absence of a T cell receptor. And it's quite striking because if you use a panel of antibodies in a spectral flow experiment, that's a multi-parameter flow experiment. And of course, what we don't use are antibodies, for example, against the T cell receptor or CD3 or GFP because we've consciously changed those. So we don't use those, but we use other markers, a whole panel of other markers, to ask whether these cells distribute in relation to the expression of these various proteins. And you can see that in the epidermis, the cells that lost the T cell receptor are now distributed based on surface protein expression in an area completely distinct from the ones that retained the T cell receptor. And actually, much the same is true for the dermis. Here are TCR positive cells missing from here. Here are TCR deficient cells that are missing from here. So now, of course, when we now reapply these markers retroactively, we can indeed confirm that, for example, these are cells where the T cell receptor expression has reduced. It's gone from red to green. CD3 has gone from red to amber. But other molecules have changed too. CD103 has actually gone up. CD69 has gone down. Likewise, in the dermis, you see these CD3 low, T cell receptor low cells are now CD69 low as well. CD103 stays the same or even goes up. And this is also seen at the transcriptomic level. You see that the cells which are derived from mice which have lost the T cell receptor either in the epidermis or in the dermis separate in this principal component analysis in PC1, which has the majority of the variants, very well based on whether or not they've retained or lost the T cell receptor. And you can clearly identify genes that are downregulated and others that are upregulated. In some cases, those changes in gene regulation correspond to changes in the surface phenotype as deduced from flow. In other cases, the correlations are less good, all of which tells us that the presence of the T cell receptor in real time is required for multiple levels of gene and protein regulation in these cells, and that the acute depletion of the T cell receptor leads to significant changes in the cell's phenotype. But what, of course, we're doing here is setting out to answer a question, which is the T cell receptor required for this lymphoid stress surveillance? And I'm going to consider three examples of this. The first is the irradiation of mouse skin with UVB wavelengths. Ordinarily, much work, some of our own and many others, has shown that this stresses the epidermis in a way that is detected by the dendritic epidermal T cells that in response rapidly upregulate 4-1BB, sometimes known as CD137, ICOS, and leads to the expression of IL-13. If you look here, what we find is in the mice in which the T cell receptor has been deleted from the skin gamma delta T cells, the upregulation of CD137 is impaired, and that of ICOS is almost completely ablated, as is the production of IL-13, not shown here. So this rapid response, which occurs within hours, cannot be made by the cells from which the T cell receptor has been removed. If we subcutaneously inoculate B16F10 melanoma, mice rapidly develop tumors. This is a challenging tumor for mice to deal with, but nonetheless, you always get a number that seem through immunological means and maybe others as well to resist the development of tumors and remain tumor-free. In mice from which the T cell receptor has been removed, this resistance is almost completely broken. And overall, in the other tumor-bearing mice, the modality of tumor size is greatly increased. When we take the gamma delta T cells from the skin, they will ordinarily induce cell death in these B16 cells. And this is greatly impaired in cells taken from mice from which the T cell receptor was removed just a week before tumor inoculation. And finally, the Aldara experiment. Here's the significant and rapid increase in ear thickness that you ordinarily see. Here's the induction of neutrophils, but in mice from which the T cell receptor has been removed, it's essentially asymptomatic. There's just no increase in skin thickness, and that's because no neutrophil influx has been provoked. In fact, when we went back and looked at mice in which Aldara had been applied, and we looked in mice in which there is a reporter, a GFP reporter for T cell receptor activation, this induces increased expression of T cell receptor signaling pathways, even though this was purported to be an entirely innate activating pathway. So the interim conclusions and perspectives at this point are that lymphoid stress surveillance refers to a rapid polyclonal response of lymphocytes to tissue challenge. It can elicit immediate effector function, for example, IL-13 and granzymes, but it can also orchestrate downstream immune responses, including dendritic cell activation, as you saw, and triggers of adaptive immunity. The response is indeed initiated by innate signals such as RAY1, as Jessica Stridge showed, or cytokines, IL-1, IL-23, likewise IL-12 and IL-18 will induce interferon gamma production. But nonetheless, there is an overt and real-time dependence on the T cell receptor that makes this surveillance distinct from that mediated by NK cells or innate lymphoid cells. So the question becomes, well, what is the mechanism? Well, one possibility that I'll refer back to later is that there is some predictable, rapidly induced ligand for this canonical T cell receptor that makes essential contributions to lesion recognition. And presumably the ligand will be different for the epidermal cells with their V gamma 5 receptor versus the dermal cells, which have got a V gamma 6 receptor or a V gamma 4 receptor. Another possibility is that there is some ligand-independent tonic signaling conferring T cell competence. But if I may stray into subjectivity, in my decades of research, it's my experience that ligand-independent mechanisms are mechanisms where we just haven't yet found the ligand, and sure enough, after a certain amount of time, the ligands are found. So I don't find this option particularly attractive, although I do acknowledge that ligand-independent signaling through the T cell receptor can be documented. But the one I want to discuss more at the moment is this notion that there might be steady state ligands that confer competence upon the T cells within the tissues that allows them to subsequently respond via innate receptors. So let's explore this a little bit more. And we can do that because of the introduction into the story of proteins known as butyrophilin or butyrophilin-like molecules that are actually members of that B7 family that I first discussed in the context of Charlie Janeway's view of integrating innate immunity with adaptive immunity through the transmission of contextual information. So this poorly studied subfamily of the B7 superfamily, the butyrophilin-like molecules, include a molecule here known as SKINT1 that we and our colleagues identified about 15 years ago, something like that. And we discovered it because we found that it was expressed on thymic epithelium, where it was absolutely necessary to engage the V gamma 5 positive progenitors of those dendritic epidermal T cells. The ectodomain of SKINT1 is shown here, superimposed upon a very famous member of the B7 family, namely PDL1. And when this molecule engages with these V gamma 5 V delta 1 thymocytes early in fetal development between E15 and E16, the consequence is to attenuate responses through the T cell receptor, as it were, passing the baton onto the cells to be able to respond to innate signals such as NKG2D or whatever. There are other molecules of this family. BTNL1 and 6 are expressed in the intestinal epithelium. They're human counterparts of BTNL3 and 8. If you look at the development of intraepithelial lymphocytes that are going to engage BTNL1 and 6 in the mouse, that too attenuates their responsiveness through the T cell receptor, by contrast to the sustained responsiveness of conventional gut T cells. And through the work on the human T cell receptor in collaboration with the Wilcox lab in Birmingham, it was clear that this reflects direct binding of this butyrophilin heterodimer, BTNL3 and BTNL8, to the V gamma 4 chain of the gut IEL that are found in the human colon. This is entirely a germline encoded motif. This is a polyclonal or a non-clonal response, depending on how you look at it. And that is actually the definition of innate immunity. So it's the use of the T cell receptor in an innate modality. But these developmental interactions cannot be the limit of the role of these butyrophilin-like molecules, because they continue to be expressed, for example, in mature keratinocytes. This is a super resolution micrograph taken by Dmitry Ushakov, showing CD3 staining, T cell receptor staining on one of those dendritic epidermal T cells, superimposed upon the adjacent staining of SCINT1 on the keratinocyte. So in published work, Duncan McKenzie used antibodies intradermally, administered intradermally. They go up to the epidermis and interrupt this interaction. And the results are striking. That constitutive T cell receptor signaling that I mentioned that's going on at steady state is immediately reduced. And by contrast, the thickness of the skin of these animals is rapidly increased. And that reflects the fact that the keratinocyte phenotype is changing. Key differentiation markers such as transglutaminase and loricrin are reduced. The cells are prone to increased apoptosis. And the ear thickening is in part because of inflammatory infiltrates of local T cells. Does this have an impact on the skin beyond the morphologic? The answer is yes, because if we measure the capacity of the animals to maintain water balance through what we call transepidermal water loss, this is grossly dysregulated in these animals, you can see here, relative to wild type. In fact, constitutive TCR delta knockout mice have increased transepidermal water loss, as is often the case in such experiments. The phenotype is even greater in the acute loss of the T cell receptor, which you see here. And this is completely mimicked if we flip the coin from SCINT1 and look now at the T cell receptor in those experiments that Miguel and Nicola were doing. You can see that again there is this rapidly induced loss of water balance. So the gamma delta T cells here, through the T cell receptor, are contributing to local physiology. These SCINT molecules are stress sensitive, so they are expressed by healthy cells, and upon stress, such as UV, are rapidly lost. So the SCINT genes are being detected on normal cells, and hence we term this normality sensing. But it's not just tonic signaling. This has a significant impact, as you can see, on barrier maintenance. So real-time dependence on the T cell receptor gamma delta at steady state is not simply tonic signaling. It's a component of tissue physiology. But having focused for a little while on this capacity of the T cell receptor, I do want to get back to this idea too, that there may still be ligands induced. More specifically, the current model for which we have clear data is that upon normality sensing, this interaction is critical, but upon stress surveillance, the molecules here may be lost, potentially handing over to the innate receptor pathways to respond to stress. It's possible that in normality sensing, there are other molecules besides BTNL molecules that regulate T cells, such as these gamma delta T cells, or other tissue-associated T cells yet to be more thoroughly studied. And there are many examples now of gamma delta T cells, but also other T cells regulating many aspects of physiology. In fact, in the August the 7th issue of Science, there is a review article on the immune system influence on physiology, relating, as does this cartoon here, to many different systems, from nutrient sensing to thermogenesis according to circadian rhythm, to synaptic plasticity and osteoclast differentiation. And the clear question now is the T cell receptor needed in all these situations? And what would be the ligands that would be driving that? Returning to the transition from normality sensing to stress surveillance, we remain alert to the possibility that there may indeed be ligands yet to be found that engage the T cell receptor, because, as I mentioned before, if we think about the Aldara system, something was driving this increase in T cell receptor signaling within hours of the addition of Aldara. So I believe that there are many aspects of T cells interacting with tissues that are driven through the T cell receptor axis via ligands that have yet to be found, but which will be extremely informative about the communication between physiology and the immune system and pathology and the immune system. I've focused experimentally on the skin, but just to say that Annette Yanka, previously in the lab, and Amber Natalini have shown that acute ablation of BTNL1 or the T cell receptor at steady state likewise impacts the gut intraepithelial lymphocytes and the gut epithelium. But interestingly enough, in humans, Robin Dart and colleagues, including Pierre Venteroux, have identified that a mutation previously described by others that naturally occurs in the butyrophilin locus impairs the capacity of the interaction with T cells that I've been describing. And these individuals have far fewer of the signature intraepithelial lymphocytes expressing VGamma4, and their phenotype is altered. And this is striking because this is linked to an association with the severity of Crohn's disease. And more recently, further mutations in this axis have been identified by Vanessa Sancho-Shimizu and her group in individuals with MIS-C, a pediatric post-COVID syndrome, in all of whom there was overt gastrointestinal inflammation. So these genetic data suggest that these lymphoid stress surveillance and normality sensing responses are non-redundant processes. And there are now many examples where the adaptive immune response is being linked to the role of sentinel lymphocytes in this way, starting with this work here on West Nile virus infection, showing that CD8 memory did not develop in the absence of gamma delta T cells. A similar story now on PubMed, on bioRxiv from Lynette Beattie and colleagues in Melbourne in the context of malaria. Similarly, in malaria, Mamedoff and Davis showed some years ago that gamma delta T cells can use MCSF to drive immune orchestration to prevent malarial recurrence. And getting back to the context of vaccination with which began this talk, we and our colleagues some years ago, looking at human responses to swine flu vaccination. Swine flu, that's what we used to think would be a pandemic. Little did we know what was coming along. But when we looked within 1 day of receiving the swine flu vaccine, the cytokine that changes most quickly in the plasma is interferon gamma, a product of lymphocytes, such as the sentinel lymphocytes that I'm describing here today. So this now leads to my third concept following lymphoid stress surveillance and normality sensing of immune integration. The notion that this connection between dendritic cells and T cells has to grow into one that includes integrated information, that includes these sentinel lymphocytes, many of which may be responding to communication with stressed epithelial cells. So the question then becomes, do these cells remain in parallel universes that rather fortunately coincide? Or is there a developmental integration that would optimize the stoichiometries required for the optimal interactions of these integrated systems? So a clue to that came a quarter of a century ago from Ishikawa's group in Tokyo, where he showed that the cytolytic and interferon gamma producing capabilities of gamma delta T cells from mice which lack alpha beta T cells in the intestine were reduced compared to those in wild type mice. You can see this is lysis of target cells induced by gamma delta T cells from wild type mice, and it's massively impaired if you take the gamma delta T cells from beta knockout mice. Well, you may say beta knockout mice lack alpha beta T cells, they're going to be all screwed up. But it wasn't true if you looked at mice which lack the alpha chain. They lack alpha beta T cells, but their gamma delta T cells were fine. Similar situation was discovered by Dan Pennington and Bruno Silva Santos when they were working with us, looking at splenic gamma delta T cells in TCR beta deficient mice. They showed less proliferation and less interferon gamma production upon stimulation, and they had an altered transcriptomic profile. But again, this was not seen in alpha knockout mice. The big difference between alpha knockout mice and beta knockout mice is that T cell development is ablated earlier in TCR beta knockout mice when the cells are trying to differentiate through what we call the pre-T cell receptor beta selection stage. In alpha knockout mice, alpha beta T cells can get through that stage and build up as double positives. They simply can't go through MHC selection into single positives. And that led to the idea that these double positive progenitors might condition the gamma delta T cells to have an appropriate phenotype for subsequent integration. And that would be lost in beta knockout mice which lacked these cells. And these ideas were picked up on and to some extent validated by Falk Wei's group within a few years of our own work shown here. But this is an issue that sort of sat to one side for a while, and we decided to revisit it because of the era of single-cell transcriptomics. And this is the work of Cara Brown and Duncan Berger. And they've done transcriptomic analyses of immature TCR gamma delta thymocytes from 6-week-old mice. This is the clustering. It obviously looks to us like an emperor penguin with a backpack on the back. And what you can see here is if you look at wild type clustering, there's considerable underrepresentation of cells here which are much more strongly represented in beta knockout mice. This is an extra control. This is B cell knockout mice. So this tells us right away that the thymocyte clusters are different according to the genotype, but this could just be trivial. We call this cluster 0. And when you look at cluster 0, it's got genes like RAG1 and the pre-T cell receptor, the pre-T alpha gene. And these are simply what we call wannabe cells. They were alpha beta T cells trying to become alpha beta T cells. They can't express a beta chain. So they express a gamma delta receptor, and they sort of look like developing alpha beta T cells. So we don't regard that as biologically particularly significant. But by contrast, look at that backpack. This is cluster 2, massively overrepresented in beta knockout mice, with a very interesting gene signature by comparison to the other clusters. And that gene signature includes MAF, SOX13, ROR gamma. These are all the hallmarks of cells which would go on to make IL-17 that are ordinarily curtailed in development during the perinatal period. And what is remarkable is if we now do this analysis in the spleen, again, we find a cluster, now it's cluster 12, which is enriched in the beta knockout mice. And guess what? It looks like a direct progeny of cluster 2 in the thymus. So what's happened is the absence of these alpha beta T cells, and presumably the alpha beta T cell progenitors, has altered the differentiation pattern of these gamma delta T cells such that a pathway which had already been attenuated in the perinatal period still appears to be producing progeny that are no longer constrained by alpha beta T cells. So let me finish by saying that immunogenicity versus tolerance was stated by Janeway and to their credit by others too, to be determined by the integration of adaptive and innate immunity mediated by this transfer of contextual information. And what I've described today is that this well-studied and essential integration of dendritic cells with adaptive immunity can evidently be regulated by interactions with sentinel lymphocytes. That is to say, immune integration has a considerably larger and more complex space than we habitually think of. In case of gamma delta T cells, these lymphocytes transition between two states. At steady state within tissue, they display normality sensing, contributing to tissue physiology with implications for tissue repair. Upon tissue perturbation, they mount lymphoid stress surveillance, empowering anti-tumor responses. But although there are myriad innate challenges that will drive this upregulation of NK receptor ligands, cytokines, etc., there is a critical dependence on the T cell receptor. And that means that these responses will be limited to those perturbations that are conducive to gamma delta T cell receptor signaling, for example, through the expression of specific ligands. These are clinically actionable observations. These give us handles by which we can regulate immunogenicity to a better degree, depending on whether we're trying to improve tumor immunity, whether we're trying to suppress autoimmunity, or trying to promote a vaccine. And these optimal integrations are in part a reflection of their tight integration during development. The future perspectives is to really, for us to understand how this gamma delta T cell receptor can pivot from these innate ligands to potentially adaptive ligands during stress responses, thereby regulating whether the status of the tissue is to maintain its integrity and its normal physiology or to mount immunoprotective responses. So 40 years on from the discovery of the gamma delta T cell receptor, we're still trying to see how it can predict through its pleiotropic multifunctional potentials when gamma delta T cells will promote immunogenicity versus tolerance. And indeed, will this be true in myriad contexts of physiologic regulation by gamma delta T cells and other T cells such as Treg cells, which have been associated with many aspects of physiologic regulation, but for which no cases that I'm aware of has the T cell receptor's role been clearly defined. So I'm going to leave it there. I gave my acknowledgements at the front, and of course, I'm happy to take any questions, which I believe are primarily through electronic means. Thank you very much.
So much sharing with us your fascinating work. We all enjoyed, and I'm sure there are plenty of virtual applauses to you. So thanks so much. As usual, this talk will be followed by questions and answers in Twitter and in Blue Sky at Global Immuno, hashtag Global Immuno. So I would like to thank everybody for joining us, and I would like to announce that our next immuno speakers will be Lympha Wang and Mate Han. So thanks again, Adrian. We really enjoy very much, and hope and I'm sure you will receive a lot of questions via Twitter and via Blue Sky.
It's my pleasure. The only displeasure I have is using any platform that has any connection to Elon Musk. But besides that, that's fine. For the
We will
for the
we
sake of
will send you.
immunology, we'll do it. Yeah.
Yeah, we will, Adrian, we will send you all the questions that we find by email. So
Okay.
just don't worry about this. So thanks so much again. It
That's
was a
cool.
real pleasure.
Thank you, Gabby. It was great to hear you, and thanks again to the Global ImmunoTalk team for all of their initiatives.